Hello dear students, today we will discuss about the histology of the nervous tissue. So we will see the description of the structure of this neuron, explanation of the section of the spinal cord, explain the neuroglial cells and its function, explain the section of the cerebrum and cerebellum. Okay, so the nervous tissue, this is how it looks, and obviously not able to really identify the neuron, the parts of the neuron, so you need to go for a higher magnification. This is at a higher focus, we are seeing this, and what you're seeing right in the center, this is the cell body of the neuron, and here this will be the axon. This is the axon here. Okay, here the other projections are the dendrites. Okay, in the center of the cell body you can see the nucleus. So the cell body is also called as soma, if you remember from the lecture we have mentioned soma. Okay, and if you see any other small nuclei, these are related to the nuclei of the glial cells, okay, of the neuroglial cells. So this is what you will see. This is how it is seen under the microscope, under very high magnification, to see the neuron and the neuroglial cells. Okay, now the section of the spinal cord. So when we study the spinal cord, you need to identify which is the dorsal and which is the ventral aspect. Okay, so this is the dorsal aspect and this will be the ventral aspect on this side. You can see from outside the covering, this will be the meninges. As you know, the meninges is also covering the brain and the spinal cord. You can also notice that there's a nerve which is entering into the dorsal horn. This will be the dorsal horn here, right? This is the dorsal horn. Okay, dorsal horn, and the nerve which will be entering to the dorsal horn will be the sensory nerve. Sensory nerve which is afferent, right? And this ganglion, this is the dorsal root ganglion. Okay, so once it enters into dorsal horn, then there are interneurons right in between them, okay, which will communicate from the or synapse from the sensory neuron to the motor neuron, right, over here like this. And then the motor neuron or the efferent fibers, they start from here, from the ventral horn cells. Okay, so from here will be the beginning of the motor nerve, which is efferent. So this is a motor nerve. And then you can see that they both meet together and it's called as a mixed nerve, right? The sensory nerve and the motor nerve meet together, and they at the intervertebral foramen, they come out as the mixed nerve. Okay, so here it is called as a mixed nerve. It will have both the fibers. Okay, what else we can see? The H shape structure which is in the center, that will be the gray matter. Okay, so gray matter, I hope you remember that the gray matter consists of collection of neurons or collection of cell bodies. Okay, then the outside, what you're seeing outer to the gray matter is the white matter. Okay, it is the white matter. White matter consists of axons or nerve fibers. Okay, consists of axons or nerve fibers. So in the center, again we can look for the central canal. This is central canal. What is the fluid in the central canal? It will be the CSF fluid. What is the lining of the central canal? It will be the ependymal cells. Okay, CSF fluid is running in the central canal also, in the subarachnoid space also, and in the central canal. Okay, CSF fluid in both. And the lining of this central canal is ependymal cells. If you remember, these are neuroglial cells, so it's called as ependymal cells. Okay, moving on. Again, the slide from the lecture slide. So you can see that this is the dorsal horn, the sensory afferent. Okay, again this is the dorsal aspect. So how to identify dorsal? If you remember, I said it is more pointed, it will touch the periphery. The ventral is more broader, okay, and usually it does not touch the periphery of the section. Here also you can see that in the ventral aspect you have anterior median fissure, and in the dorsal aspect it is called as dorsal median sulcus. Okay, fine. Then here there is neurons, interneurons present in between the two. Here the motor efferent, which is the one which is coming out, can be seen. Okay, motor efferent. Then the gray matter, which is the H shape over here, and white matter, which is outside. Okay, gray matter and white matter is outside. Gray matter is inside the H shape, and then white matter is outside. The central canal is noticed here. Okay, here also we can notice the lateral horn. This is called as the lateral horn. So there are three horns here: the dorsal horn, which is sensory, then the ventral horn, this ventral horn here, which is motor here. Okay, sensory, this motor, and the lateral horn is only seen in the thoracolumbar region. It is for the sympathetic supply, okay, related to the autonomic nervous system. Okay, so the lateral horn, it is not seen in all sections of the spinal cord. Okay, it is related to the autonomic nervous system. Okay. Further, we can also notice something called as neuropill. Okay, so neuropill, it is the processes of both the neurons and neuroglial cells. Okay, so this is a place where you can see the neuropill, like the process of the neuron and the neuroglial cells will meet in this place, so that is called as the neuropill. Okay, just want you to know the name of this area. And again we can see some neurons. Okay, the nucleus of the neurons, and you can see the dark staining by the Nissl bodies. Okay, Nissl bodies are present within the nucleus. Okay, then this is also the gray matter area, and this is the white matter. So the previous slide in which we saw the H and the area around the H, which is the white matter, this is a higher focus just to see the structures more clearly. The H, which has these cell bodies, okay, collection of cell bodies, which is the gray matter, which we have told. And white matter, here in the white matter you will not see any cell bodies. Okay, only axons are seen. These are the axons. Okay, then the difference between ganglia. Okay, so ganglia, we have sensory and motor ganglia. The sensory ganglia, usually they are in groups, and they are of the similar size. They are made up of pseudo-unipolar neurons, and okay, if you look at the photo, it's more darkly stained here, and around the neuron, these what you see SC, this is the satellite cells. Okay, satellite cells is the neuroglial cells related to the peripheral nervous system. Okay, neuroglial cells related to the peripheral nervous system. So we had in peripheral nervous system two neuroglial cells. One is satellite cells, the other one is Schwann cells. The satellite cells, they help in insulation around the ganglia. Okay, so the satellite cells are present here. Fine, and you can see that Here this is a nerve fiber which is noticed, okay, and they are more rounded in shape, okay, rounded. Coming to motor, they are more scattered, okay, and the shape is of different sizes, and it's angular in shape, angular, like it will because of the cut, and they are made up of multipolar neurons. So here the neurons are multipolar, okay, and obviously by the slide you notice that it is quite lightly stained, okay. Here also there is satellite cells. Satellite cells are seen around, okay, and around the ganglia. Okay, next, the nervous tissue histology mainly consists of neurons, which are the true conducting cells of the nervous tissue, okay, and the neuroglial cells, okay. So these are the neuroglial cells in the CNS. The CNS neuroglial cells are 4 astrocytes. They are star-shaped cells and they are situated near blood vessels. They have a lot of functions. One of the important function is blood-brain barrier. Oligodendrocyte. Oligodendrocyte helps in myelination. Microglia, micro, the word micro because it is also derived from the macrophage system. It helps in phagocytosis, one of the smallest cells. Then you have ependymal cells which secrete CSF. So they are seen along the lining of the cavities, that is the ventricles and the central canal. In the PNS we have 2 cells, that is the Schwann cells. Schwann cells are just like oligodendrocytes, okay. So they will help in myelination along the nerve fiber. And satellite cells are seen around the ganglia. So both of them help in insulation, okay. Schwann cell also, the reason why they do myelination is they help in insulation, so that there is no so that the nerve conduction is in one direction, okay. Okay, here we can see the astrocyte. This is an astrocyte, yeah. Okay, here the one which is marked in V is actually the veins, okay. So since we told that the astrocytes are related to the blood vessels, so this is an astrocyte. This is one astrocyte, this is one, and this is another one. There are 2 astrocytes over here, okay. So 2 astrocytes and the blood vessel profiles are seen in Golgi stain. They have used a special stain. Astrocytes have processes that may be long, slender, and relatively unbranched, or short and highly branched, okay. So that's astrocytes. Hopefully in pathology you will also study some tumors related to these cells, okay. So that will be called as astrocytoma. If there is any tumor, it's called as astrocytoma. So the word astrocytes, you should remember these cells, okay. Coming to ependymal cells, they are related to the CNS, and they line the ventricles and central cavities of the brain and the spinal cord, and they secrete CSF. They are ciliated to help circulate CSF, okay. So these are ependymal cells. Like it's like simple cuboidal with cilia, simple cuboidal to columnar with cilia, okay. So these are ependymal cells, okay. So here we can see this is a ventricle, okay, and what you see in N, that is a neuropill, which we have already described before. So it's a meeting of the processes of neurons and neuroglial cells. And E, this lining, these cells which you're seeing here, these are the ependymal cells, okay. So we can also see some cilia over here, so that's why it's the ependymal cell. So they secrete CSF. Here we can see a myelinated nerve. The myelinated nerve has Schwann cells, okay, Schwann cells for myelination, right. And this is a place called as node of Ranvier, okay, between yeah, if you've seen the diagram, the schematic diagram, you notice that there is nodes in between the myelination, okay. This is a peripheral nerve connective tissue. So the names you should know. The outer covering, okay, outer covering is called as epineurium, which covers the entire nerve. Okay, so the entire nerve is covered by epineurium. Epineurium. Then there is perineurium. Okay, next is okay, this is perineurium, which is covering the nerve fibers. Okay, so that's so the epineurium is covering the entire nerve like this. Okay, for example, the perineurium is covering the nerve fibers like this. Okay, and then there is endoneurium. Okay, there is endoneurium, which is supporting every individual nerve fiber. Okay, so it's not shown here, but just understand there's endoneurium, which will be supporting each nerve fiber. Okay, what you see, A is artery. Okay, this is an artery over here, artery, and this is a vein, right? And this is a nerve fiber, what you're noticing. Okay, so this is the covering of the peripheral nerve connective tissue. Now coming to histology of the cerebral cortex, there are 6 layers. The 6 layers are from up to down. You can see that it is the molecular layer. The second layer is called as external granular layer. Third is external pyramidal layer. Fourth is internal granular layer. Fifth is internal pyramidal layer or ganglionic layer. And the last one is polymorphic or multiform layer. Okay, so if you will notice here, let's go to this diagram. So this is the first layer, which is molecular layer. Here you will not see any cells, that is the pyramidal cells or the granule cells. Okay, then the second layer is external granular layer here. The third layer is external pyramidal. So how to notice the pyramidal? Look for the triangular shaped cells. If you see triangular shaped cells, that means they are pyramidal cells, and that is in the third layer. The fourth layer again will not have pyramidal cells, so they are called as internal granular layer. The fifth layer will have large pyramidal cells. Okay, so as you notice here, there are large pyramidal cells. And the last layer is the sixth layer, so it's beyond the fifth layer, so like that you can remember. Now let us see how it is seen again. Here I will show you, this is the first layer, okay, molecular layer. Okay, and this will be the third layer, which is the external pyramidal layer, because I can see the dark pyramidal cells. That means this will be the second layer, external granular layer. Okay, then here, okay, these are all the fifth layer, okay, where I can see the internal pyramidal cells. The cells are large. Okay, here you can see that there are large pyramidal cells. Okay, so they are like this, large pyramidal cells. Okay, so then that means this is the sixth layer. Okay, multiform pleomorphic layer, and this will be the fourth layer. Okay, so again the first layer is molecular layer, second layer external granular layer, third layer external pyramidal layer, small pyramidal cells, fourth layer internal granular layer. The fifth layer is internal pyramidal cells, which is also called as Beth cells. It's very, very important. It's the beginning of your corticospinal tract or pyramidal tract. The word pyramidal starts from here because it's pyramidal cells. Okay, so when you hear the word corticospinal or pyramidal, okay, corticospinal tract, pyramidal tract. Okay, so they all start from here at this layer, from the fifth layer. Okay, so pyramidal tract, it starts from the fifth layer of the motor cortex, area number 4, and that's where it begins, and then it goes down. Okay, Fine. So here there's a higher view, higher magnification, in which you can see the pyramidal cells. Okay. So if these cells or the layers are asked, like, I hope you will be able to identify them. Now coming to cerebellum. Okay, so this is cerebellum. Okay, so cerebellum, if you see the architecture, it has folia and fissure. Folia means these, when it goes like this, it's called folia, and then when it goes deep inside, it's called as a fissure. So this arrangement, so the folia is the one which is extending outwards, fissure when it goes within. Okay, so this is folia and fissure. This is the arrangement of the cerebellum. It's different from the cortex, cerebral cortex. Cerebral cortex has sulci and gyri. Okay, now if you notice the general pattern, outside you can see the pale color, the pale pink color. So that is the molecular layer, right? We'll see in the next slide in more detail, but just to have an overlook. So this is the molecular layer. The Purkinje cells are not seen clear. Purkinje layer will be here, and then this is the granular layer where it's dark. Okay, the dark layer will be the granular layer, correct? Okay, now deep to that again you can see the pale pink, right? So that is nothing but the white fibers. That is the white fibers. Okay, so if you notice, like, let me take one example. So here this is the molecular layer, this is the Purkinje cells, this is the granular layer inside, then this is white fibers. Here again on the other side it is. So this is molecular, this is Purkinje, this is granular, and white fibers in between. Okay, so what is white, about the white fibers, what do you know? The white fibers, okay, they form the central core, okay, and they have a characteristic tree-like pattern. It's called as arbor vitae. So these white matter is made up of fibers either entering, that is afferent, or leaving, which is efferent, to the cerebellum. So they have three bundles. They make three bundles, that is the superior, middle, and inferior cerebellar peduncles, which connect with the midbrain, pons, and medulla. Okay, so that's about the white fibers. Okay, now let's see the next. You can again notice, so this is a higher magnification. Sorry. Okay, so remember for the cerebellum, I remember that I had told you that there are 3 layers. For the cerebrum it is 6 layers, and for cerebellum it is half of it, 3 layers only. Okay, so this is the outer molecular layer which you're seeing. So you will not see, it is not darkly stained, it is pale. Okay, that's why it's got this color. Then this is the Purkinje. This is the Purkinje, okay, right in the center here. And second layer is the Purkinje cells, and then the inner one is the granular layer. Okay, it's darkly stained. It's densely packed with lots of cells. And if I ask you what is this here, again the light pink over here, that will be the white fibers, correct? It is the white fibers. And then what is the details about the molecular layer? The molecular layer is made up of interwoven fibers, mainly only fibers, and the granular layer is made up of large number of closely packed microneurons, lots of cells here, granules. That's what gives this appearance. Intermediate Purkinje cell layer is made up of single layer of large flask shaped Purkinje cells. Okay, so the afferents which reach the cerebellar cortex, are usually conveyed to the cerebellar nuclei via the axons of Purkinje cells. Okay, so now if there is any afferent which is coming, okay, if it comes to the cerebellar cortex, if it's coming here to cerebellar cortex, then it is going to the cerebellar nuclei. Now where is the cerebellar nuclei? Obviously it's situated inside. Okay, it will be situated within. So it is traveling through the neurons of the Purkinje cells. Okay, So this is the function of the Purkinje cells. It carries the afferents which reach the cerebellar cortex, okay, to the cerebellar nuclei via the axons of Purkinje cells. And here you can see the shape of the Purkinje. Okay, they are called basket shaped cells. Okay, so this is the shape, and this is the axon which we were talking about. So this is the axon which is extending in the molecular layer. Okay. Okay. What are the nuclei? Just quickly I will tell you the nuclei of cerebellum. So we have it's called vestigial, then you have globose, then there is emboliform, and then there is dentate. Okay. So the dentate is related to the most recent phylogenetic development. The uh, the vestigial is related to archicerebellum, the old one, archi. Okay. Then the globose and emboliform is associated with paleocerebellum. They both are related to paleocerebellum. Okay. This is dentate is related to neocerebellum. Okay. There is a mnemonic of remembering these names. Okay, we say that dentis. Okay, dentis. Eat good food. Okay, so D dentis. That is this is from how is the relation from the sequence is from medial to lateral. Okay, what we have said from vestigial to dentate, from medial to lateral. Okay, so dentis, the lateral most, then a dentate, then eat, the emboliform. This is for emboliform. Globose, that's G over here, globose. It's like glucose, globose, remember, like this. And vestigial, vestigial. Okay, that is good food. Okay, so dentis, eat good food. Please remember the name of the nuclei, it's very important for you. However, I will not be able to ask you in histology, but it's something which is important to remember. Okay. Finally, we have finished. I hope it was easy, and if you have any doubts, you can ask me during the practical sessions. Thank you. All the best for your exams.